Hello and welcome to Clinical Path. Today's topic is the structure of the heart at the level of cells, organelles, and proteins. In the previous lecture we noted that structure is a very comprehensive term in understanding how an organ is built. It starts with the regional architecture, which is important because organs affect each other because of physical proximity. And for the heart, this would be the thorax. We then look at the surface of the heart and then the internal structures. From that point then we go to higher power, and tissue refers to the architecture of how the cells join to each other. From the individual cell we take a look at its ultrastructure of organelles, which are built of proteins. So in today's lecture we're going to start at tissue and we're going to follow that through to proteins. In the previous lecture, we talked about how the gross anatomy of the heart can be seen in higher power at the tissue level. So here we have a short axis view of the heart by echocardiogram, and you can see we've taken this portion of the left ventricle, which we've blown up to see how the individual cells are put together. And when we talk about cardiac muscle, we can see that it forms an interdigitating mesh. By forming this connected mesh, it creates a chamber by which we can see a single pulse of blood, which is going to be our stroke volume. It functions as a displacement pump. When we look at the key elements of this design, we start here by noticing the cells actually join end to end. And you can see that the structure that joins each cell in this axial fashion here is going to be our intercalated disc. Next, we consider that the cells have to have a way of coordinating the signaling, and it does this through numerous ion channels which are found in the cell membrane. And then last, these coordinate the actual mechanical contracting cell, and the contraction occurs through all of the proteins that are organized into sarcomeres. It's useful at this time to compare the design of a cardiac system to skeletal muscle, because skeletal muscle is designed to go across a joint and insert through tendons into the bones and contract in a single pulse. And it's a voluntary contraction, so it's got to have a nervous system input. These skeletal muscles are arranged in fascicles. So a fascicle is a bundled group of cells, each of these a fiber. And a fiber actually is a myocyte. And if we look within that myocyte, we can see all of the sarcomeres joined end to end in fibrils. And note now this has to be under voluntary control, so it has to be a nervous system control. The nerves come down and join to that fiber as a neuromuscular junction. Compare that now to how the smooth muscles are organized. These now are joined as loosely arranged individual cells. Nerves are going to come in and not as neuromuscular end plates, but just loosely into the interstitial, and then they diffuse their signal out in a paracrine-like fashion. Also, motor coordination can occur through hormones. The signaling is passed from cell to cell by numerous gap junctions. Skeletal muscle has myofibrils. There are motor end plates, also known as neuromuscular junctions. The speedy signal of the calcium is enhanced by the presence of T-tubules, which come from the sarcolemma. And then there has to be special cells that join the muscle cells to the tendons, so there are myotendinous junctions. And that's very different than smooth muscle cells, because these have no motor end plates. There are numerous gap junctions. There's no regular order to how the cells are arranged together. And then last but not least, smooth muscle cells, in addition to contracting, also regulate the extracellular matrix. And that's going to be really important when we go to many diseases of blood vessels. Well, let's look at the intercalated disc. The cells are joined end to end by this interdigitating architecture. And there are two key elements to consider in this. The first is that there have to be really rigid structures so the cells don't pull apart in contraction. And these are going to be our desmosomes. And desmosomes consist of key joining proteins such as desmoglein, which go through and connect into the underlying cytoskeleton so that we have great strength through the entire cell. And classically, the cytoskeleton it's going to join to are going to be intermediate filaments such as desmin. The second key element are going to be gap junctions, very thin membrane areas that allow for free flow of electrolytes. The depolarization signal can freely flow through these gap junctions, and that's what's actually going to create our depolarization signal. When we consider this architecture, we can see that it actually has some really important clinical relevance. The first is going to be that there's no regeneration of cardiac myocytes. And that makes sense because a cell is only functional if it's connected to end with its neighbors and can communicate through the gap junctions. Just creating another cell that isn't connected wouldn't actually help. These cells, when they need to increase their output, do this by getting bigger through hypertrophy, not hyperplasia by increasing their numbers. And then the last is to think about the gap junctions, because the gap junctions can propagate both directions. This can cause reverse flow of the signal and is one of the causes of cardiac arrhythmias. The ion channels are different between conducting or pacemaker myocytes and contracting myocytes. Start by thinking about how they work in the conducting cells. First of all, there is a resting potential of minus 60 millivolts, and that's going to be set up by two different channels. The first is going to be our sodium-potassium ATPase. That's the heavy working pump. The second is an open potassium channel. The open potassium channel has two different forces working on it. The electrochemical force, where the negative charge is drawing it in, but then it also has the concentration gradient drawing it out. When we take a look at the balance of the ATPase and the open potassium channel, it settles at a resting potential of minus 60 millivolts. And that's an important number to note, because when we talk about the fast sodium channels that are going to be very important for signaling in the contracting, the fast sodium channels actually activate at minus 70. They are not involved in any of the functions of the conducting pacemaker cells. From this resting minus 60 then, what makes it a cell with automaticity is going to be the pacemaker or funny sodium channel, because this allows for a leak of sodium in. And that chronic leak of sodium is going to change the internal potential such that when it gets to minus 40 millivolts, it opens the calcium channel. And that then allows the inflooding of calcium that's going to create depolarization. And then charge is balanced by the potassium outward or delayed channel that's going to establish neutrality again. So as we follow what this looks like electrically, we can see that there is a maximum negative potential here at minus 60 millivolts. And with the pacemaker channel, there's this gradual change in potential to minus 40, which becomes our activation potential. This correlates with the drifting upward. And at minus 40, we're going to activate our calcium channel, and then we're going to restore our balance here through our potassium channel. These are given numbers. The sodium drift is called stage 4, and the calcium here is stage 0, and then over here we have our potassium efflux. That then is the cell and the channels that are going to create the automatic depolarization signal. Now note the difference that we come to in a contracting myocyte, because now between the potassium-sodium ATPase and the open potassium channel, the resting potential is minus 90 millivolts. Now when we get the depolarization signal, and you notice we don't have a pacemaker channel, now that signal is going to be coming from the adjacent cell. all that influx of sodium creates the signal. And when that signal hits minus 60, that's now going to open my fast sodium channel. And when the changes from all that influx of sodium now gets to minus 40, that's now going to activate my voltage-gated calcium channel. In comes the calcium, and now I've created my calcium signal for contraction. Take a look at what that looks like electrically. Now you can see that our maximum negative potential starts at minus 90 millivolts. Our activation threshold potential is still the voltage-gated calcium channel, so that's still at minus 40. And so we can see coming along here, it's in steady state. We get the depolarization signal, and then there's the calcium channel, and there's my potassium restoring balance. This becomes our resting state of stage 4. This is stage 0, where my fast sodium channels open at minus 40. This is now going to open my calcium channel, stage 2, and then we open the delayed potassium channel and restore electrical status. We've gone through how we create a signal, and we've gone through how we create the calcium signal, which is going to be our signal to the sarcomere. So how then does the calcium signal start contraction? Let's blow up a membrane and give ourselves our voltage-gated calcium channel. When the signal comes along, we get this massive inpouring of the calcium. And that calcium is actually going to go and signal a receptor on the smooth endoplasmic reticulum called our ryanodine receptor. And when that ryanodine receptor receives the calcium signal, it allows for this massive outpouring from the smooth endoplasmic reticulum of calcium. This is calcium-induced calcium release. So what we've done basically is we've just amplified the calcium signal. And that calcium is now going to come down and signal the troponin C protein, which is going to open up and allow for the tropomyosin to open the actin active sites. The calcium is going to be pumped back into the smooth endoplasmic reticulum by the smooth endoplasmic reticulum calcium ATPase. And this is going to be regulated by an inhibitory protein, our phospholambin. And then the signal is finally shut down because there are membrane pumps, sodium, calcium exchanger, which is usually written as a sodium calcium exchanger. And then there's a small, much less important calcium ATPase. And when we correlate what this looks like to our signal, we can see then here's our electric signal here coming through. There's our calcium voltage-gated channel signal to the calcium. Calcium is a signal that gets amplified from the smooth endoplasmic reticulum, opening up the conformation of troponin C and actin-myosin interaction. How does the calcium signal actually cause the sarcomere to contract? Well, the first thing we need to think about is we should build a sarcomere. And a sarcomere is defined as the space between two Z lines. And built into these strong structures are going to be our actin molecules. And sitting inside these actin molecules are our myosin fibers. And it's going to be the interaction of these two fibers that's going to create contraction. These require some organization. It's too damaging for the myosin to simply pull against the Z line. So the myosin is buffered by a very elastic protein anchoring it to the Z line, which is our titin, the largest protein known in the body. And the second thing is the cell actually has to be anchored into the extracellular matrix. And the protein that's going to do this is going to be dystrophin, which anchors into a dystrophin membrane complex, and from there out into the extracellular matrix through proteins like laminin. The regulatory protein complex that regulates the actin-myosin interaction is going to start with our tropomyosin, and it wraps along all of the actin to cover the active actin sites. And tropomyosin itself is actually regulated by troponin. And it's going to be the calcium signal that comes in, binds to the troponin, and allows that for a conformational change in the tropomyosin that opens up the active sites for actin that the myosin head can bind to. So we can see then we have contractile proteins, which are going to consist of our actin, myosin, and the regulatory proteins of tropomyosin and troponin. And then you can see we have structural proteins such as dystrophin and titin. The last piece of this discussion then has to be, how do the actin and myosin heads interact? So if we take a standard Z line here and put actin on it, and we draw in our myosin, myosin actually has heavy chain heads, and normally these are prevented from interacting with the actin by the tropomyosin that's under regulation. When that tropomyosin goes through its conformational change from the calcium signaling, it actually allows this head to touch the active site of the actin. And when that myosin head actually touches the active actin site, it causes ATP hydrolysis that causes a flexion of the myosin head itself, and that gives us sliding of the actin site. The ATP then releases the actin-myosin site, and we're now left with an unbound myosin ready to reattach to the next site. And so we get this attach-rea site, which gives us this sliding motion of the actin. One of the clinical correlates that helps you think about this is what happens when we get myocardial infarction. What can happen then is there's a complete lack of ATP. And when we take a look at the myocytes present in that dead muscle, we can see these contraction bands that form. They represent the intracellular calcium from the ischemia are going to induce maximum contraction, but it's hypoxic and there's no ATP, so that they can't release the contracting heads. And so we see these contraction band necrosis when the myofibrils contract, but can't release. In summary then, we should be able to be clear in our mind about the architecture of the cardiac myocytes. Remember, these are interdigitating and end-to-end. And this is important because it's that continuous mesh that creates the positive displacement pump of the stroke volume. The second thing to remember is there are two very different cell types. We have the conducting cells, which have automaticity, and then we have the contractile myocytes that contain numerous sarcomeres for the sliding action of actin to myosin. When we talk about the actual structure of the myocyte, remember there are going to be three key elements. The intercalated discs that actually contain the desmosomes so they don't pull apart, and the gap junctions so that the ions can come through as the depolarization wave. There are going to be the ion channels, which is how we're going to create the signal of calcium in the cell. And then we're going to have the sarcomeres that form the actual contractile unit. The signal is a calcium signal amplified by calcium-induced calcium release. And then the last element is going to be contraction, in which the calcium affects the troponin C, which affects the tropomyosin, which allows for the actin active site to interact with the myosin heavy chain head. That brings us to the end of this topic. The next topic to follow is going to be regulation of this contractility. See you next time.